Hi everyone, this is Miss Snook, and today I'm going to show you how to do Lab 5.16, the lab on root fixation. So in this lab you're learning about bacteria that form what's called a symbiotic relationship with plants to help plants convert nitrogen into a usable form in order to grow. So the first thing you need to know is what is a symbiotic relationship, and basically it's a mutual relationship between two organisms where one benefits the other and the other one benefits the other. So it's like I'll scratch your back, you scratch mine. The second thing you need to know is what is nitrogen fixation, and nitrogen is a gas in its normal standard temperature and pressure, and it needs to be formed into a compound in order for a plant to use it, and most notably those are nitrates, and one of those is ammonia. The bacteria you're going to be learning about today is called Rhizobium, and it lives in the roots of plants. So if you click on this little plus sign right here, it'll zoom in on the roots, and it'll zoom in even further here to show you these little bacteria. They are a rod-shaped bacteria, and they live in the soil. This is a picture of a root with the Rhizobium bacteria on it, and you'll notice that it has a pink color to it, and it explains to you that when the Rhizobium is pink, what's happening is it is actively taking that nitrogen gas and forming it into a compound so that the plant can use it for growth. When it is green, it means that it is reproducing, and what's important to note is that it can't do both activities at the same time. On the next slide, you'll see a cross-section of a microscopic view of the root that the Rhizobium live in, and if you zoom in close enough, what you'll see are cells inside of the root that have Rhizobium present. Those are the purple ones that you see here, and then the cells that don't have the bacteria living in them are in blue. You're going to need the student guide for this lab, which is located on page 5, so go ahead and open that up. That will give you the directions of what you're supposed to do in the virtual lab, which is on the next page. You're also going to need the lab report, which is located up in Doc Sharing, and the lab report is called Lab 5.16, and it happens to be the first one listed in this particular example.
Have a bean. Have a bean.
And that was my son coming in to interrupt while my door was closed and I was recording, so I apologize for that. Now you're going to open up the virtual lab, and you click on the virtual lab. It will take you outside of the K-12 website, and you would need to view the tutorial to see how to use the tools. But I'm going to go ahead and begin the lab to show you how this is done. I'm going to go ahead and select my gloves. And if you have your student guide open, you'll notice that the introduction tells you to go ahead and do the tutorial so that you can learn how to use the tools. But you'll be able to see how to use the tools as I walk you through this lab. So part A is you're going to first observe the roots. And what I recommend you do is you zoom in on these. So zoom in as far as you can. You're going to come over here and pick up your hand lens. So you just hover over the hand lens and your glove is going to change a shape. You click on that to pick it up, and then drag it over here and take a look at these roots. When you're done inspecting the roots, you're going to record your observations in the lab report in the appropriate area. You can either give a very detailed description of what you saw in words, or you can also draw a picture and label it as best as you can with what you see in this picture. If you're not sure of what you're looking at, you can use words to describe what you're seeing. When you're done inspecting the bean plant and the radish plant, go ahead and put down the hand lens and fill out your lab report for that section. The next step is you're going to do part B, which is actually observing the bacteria in one of these nodules. So you're going to come over here and grab your scalpel, click to pick it up, And you're going to come over here, and this big nodule that's right here that I'm pointing to, you're going to go ahead and click on that. It's going to cut it off of the plant. And you're going to go ahead and put your scalpel down, come back over to the nodule, pick it up, and move it up here to this dissection plate. Next, you're going to come back down and grab your scalpel, and you're going to cut your seed in half, your nodule in half by just clicking on it. You can put your scalpel down, and you're going to pick up your hand lens and come back up to it and observe what you see. You're going to sketch what you see in the lab report. You're more than welcome to hand sketch it and label it, or you can use the drawing tools in your word processing program or open office program. So draw and label what you see. You might need those parts from back in the actual lab. Next, you'll put down your hand lens, and we're going to move to the microscope. I'm going to zoom out a little bit for the microscope so you can see all the things that we need. In order to remove the microscope cover, you're going to click on it, and you're going to throw it away, the cover. I don't know why it requires you to throw it away. Oh, you can put it off to the side. Don't throw it away. Alright. Next, you're going to clean with some lens paper your eyepiece. So you're just going to rub that on the eyepiece. You're not going to hear anything while you're doing that. And you'll go ahead and throw that in the trash. It's going to ask you if you want to. Just hit yes. Next, you're going to pick up your slide. You're going to place it on the stage of your microscope. And you're going to turn on the light for your microscope. And you can adjust the lighting here by clicking on this draggy bar down at the bottom. And you can adjust where the stage is by holding your mouse and clicking over here. So the next thing that you're going to do is you're going to adjust your focus. And this is the hard focus up here. So you're going to click on it, and you're actually going to move your mouse up and down. Moving your mouse up and down is what's going to focus this. Oops, I broke my slide. That's great. I broke my slide because I was on 40. You should have it on 4. So you're going to adjust it by going like this, and then do your fine adjustments down here. So you're going to hold the knob, but you're just going to move your mouse back and forth. And you're going to draw what you see. You can adjust the magnification by coming over to the lenses here and holding your mouse down and moving it till you feel like it's clear enough to draw a picture. Fine tune it here. It'll warn you if you've moved it too far, like it warned me, and I told it to keep adjusting. Alright, and that's as close as you can get, is at the 40 times. So you're wanting to draw those rhizobiums and label each one of the parts just like you saw in the lab. When you're done, there is a bottom part to the lab report that asks you a few questions. The first question is, 2 new species of plants were introduced into an area that was low in available nitrogen compounds in the soil. 1 formed nodules and 1 did not. Which species would you reasonably expect to grow more robustly? So you're going to pick which species you think is going to grow the most and explain why. The next question is, how does the location of the nodules relate to the function of the nodule? And so you'll want to read about that in the lab and go ahead and give an example. And last but not least, the last question is, this diagram shows 2 groups of plant cells, one with rhizobium and one without the bacteria. Label which is which and explain the obvious size difference using observations from Part C in your lab. And the answers to those are also in the reading in the lab inside of the K-12 website. When you're done with this lab, it goes in the Dropbox for Lab 5.16. If you have any questions, please let me know. Thanks for watching. Bye.